A recent study conducted on mice has shown promising results in reversing amnesia caused by head injuries. Greetings to everyone. In today's video, we will be discussing a fascinating discovery made by researchers at Georgetown University Medical Center and Trinity College, Dublin. Their study, which was published in the Journal of Neuroscience, reveals that amnesia caused by head injury is not an irreversible condition associated with neurodegenerative diseases. In fact, the researchers were able to reverse the memory loss in mice and restore their ability to recall lost memories. Initially, they used lasers to activate specific engram cells, but they are currently exploring non-invasive techniques to communicate with the brain and reset it to its previous state. So, without further delay, let's dive into our discussion. Crucially, the scientists made a significant discovery regarding the memory loss associated with head injuries. They determined that this loss of memory is not an enduring pathological occurrence caused by a neurodegenerative condition. In fact, the researchers were able to effectively restore the lost memories in mice, offering the possibility of reversing cognitive impairment resulting from head trauma through clinical interventions. By altering the synaptic functioning in the brain, the researchers at Georgetown University discovered that repeated head impacts can have a detrimental effect on memory formation and retention. However, their latest study demonstrated that it is possible to reactivate forgotten memories in mice that were previously affected by head trauma. According to Mark Burns, PhD, the senior investigator of the study, and a professor at Georgetown University's Department of Neuroscience, there is optimism that our research can pave the way for the development of treatments that can restore cognitive function and normalize the head impact brain in individuals suffering from memory impairment due to repeated head impacts. Dr. Burns also serves as the director of the Laboratory for Brain Injury and Dementia. The researchers conducted a recent study in which they imparted a fresh memory to 2 sets of mice through training them in an unfamiliar test. For a duration of 1 week, the first group of mice was subjected to a series of mild head impacts at a frequency comparable to the exposure experienced in contact sports by humans. Conversely, the second group, serving as the control, did not receive any impacts. When the mice that had been impacted were tested 1 week later, they demonstrated an inability to recollect the newly formed memory. Burns explained that the majority of studies conducted on this subject have focused on individuals with chronic traumatic encephalopathy, CTE, a progressive brain disorder observed in individuals who have experienced repeated head trauma. In contrast, their objective was to investigate the alterations that occur in the brain as a result of the frequent but less severe head impacts commonly encountered by young football players. In the realm of college football, researchers revealed that players endure an average of 21 head impacts per week, while defensive ends face a staggering 41 head impacts weekly. To emulate the weekly exposure experienced by college football players, the study subjected mice to a comparable number of head impacts. It is important to note that each individual head impact was exceptionally gentle in nature. By utilizing genetically modified mice, the scientists were able to observe the specific neurons responsible for the formation of new memories. Interestingly, they discovered that these memory neurons, also known as the memory engram, were present in equal measure within both the control group and the experimental group of mice. In order to comprehend the physiological changes that occur in memory, Daniel P. Chapman, PhD, the primary author of the study, explained, "Our ability to associate memories with specific locations is a result of the reactivation of our memory engrams when we are in or see a photo of a particular place. To identify the distinct markers of an activated neuron, we investigated the engram neurons. While the control mice were able to activate their memory engram when they were exposed to the room where the memory was initially formed, the mice that experienced head impact were unable to do so. This inability was the cause of their amnesia. By utilizing laser stimulation on the engram cells, the researchers successfully reversed the amnesia in mice, enabling them to recollect the memories that were once lost." However, Burns clarifies that the invasive method employed in this study cannot be applied to humans. As a result, the team is now exploring various non-invasive techniques, in order to signal to the brain that it is no longer at risk and to initiate a period of plasticity that can restore the brain to its previous state. Alongside Burns and Chapman, Stefano Vicini from Georgetown University, as well as Sarah D. Power and Thomas J. Ryan from Trinity College, Dublin, Ireland, are also included as authors. The research conducted received assistance from the Mouse Behavior Core within the Neuroscience Department at Georgetown University, as well as grants from the National Institutes of Health, NIH, and the National Institute of Neurological Disorders and Stroke, NINDS, under the codes R01NS127370 and R01NS121316. NINDS also provided support for F30NS122281 and the Neural Injury and Plasticity Training Grant, which is located in the Center for Neural Injury and Recovery at Georgetown University under the code T32NS041218. Additionally, seed funding was obtained from the CTE Research Fund at Georgetown. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.